Welcome everyone to the Department of Genomics and Immunology, established at the Sri Madhusudan Sai Institute of Medical Sciences and Research. My name is Vamsi Yenamandra and I am currently serving as the director for this department. I'm a dermatologist by training and I'm also a scientist who has extensively worked on rare diseases. My focus has been developing diagnostics and therapies for these particular incurable diseases across the globe. I have previously worked at AIMS New Delhi and then Netherlands and at Stanford University, USA, where I have led the first topical gene therapy for a rare disease called epidermolysis bullosa. Coming back to India, I would want to develop more accessible therapies and affordable therapies for these patients back in our underserved populations in the country. So I would like to show you around the department right now. This is where the department begins with. This is where the admin receives the samples that are referred across the hospital. Sample entry gets done and barcoded so that patients beyond this point becomes de-identified, thereby protecting the privacy of the patients. This is the office space where students and the faculties from different departments do their independent works, where we collaborate together and to make meaningful projects that are useful for the community. This is the student sitting area where we currently have two PhD students from the university, from the life sciences department, where we are trying to integrate research, basic research with our clinical research so that it benefits patients at large. Now we will be walking through the different components of the lab. There are 3 independent labs that are there, each segregated for the work that they do. The first component that we are going to see is the DNA isolation room, where the samples that have been initially barcoded over there, the DNA will be separated from these samples in this particular component. We have students already working, so the students will be going ahead and explaining what equipments and the work that they're doing.
The DNA purification process mainly involves cell lysis and washing out methods where we remove unwanted materials and elution where we collect the purified DNA. There are multiple methods to isolate DNA. In our lab, we are using salting out method, magnetic bead method and spin column method. The quality of the purified DNA is very important as it forms the very foundation for reliable molecular research, diagnostic processes, and also for the precision medicine. This is a working station for the isolation of the DNA. Here we have spinners and vertexes which are used during the isolation of the DNA. And we also have a hot water bath, which is very important to maintain the temperature during the isolation of the DNA. This is a gel electrophoresis which we use to check the quality of the DNA. Once the DNA quality and quantity are confirmed, it is ready for the downstream processes.
Once the DNA is isolated, this room caters to library preparation, wherein we segregate each samples, plug them together so that the sequencing happens.
So in this library preparation step, we make it sequence ready so that we can do sequencing for the extracted DNA. For example, when you look at this instrument over here, this is an iBright i500 imaging system where we see the amplification which has happened in the DNA. So for example, if this is the gel and we place it in this drawer and we press, you can see that it visualizes the amplification which has occurred. We can see the amplification has occurred in all the wells, but in the last well is where a proper amplification has occurred. This is a quality control step where we understand whether our amplification has worked or not, so that we can move on to the next steps.
This part of the lab is after the library preparation is done, and this is the heart of our genomics lab, where we have a sequencer which is called MiSeq i100. So recently Illumina has launched this, and this is our first installation in the country where we have an automated sequencer and along with a computational server,
which is primarily focused on MiSeq i100 Illumina platform. The lab was designed as a translational genomics facility which is capable of doing research and clinical diagnostics and precision therapies. So the idea, the core strength of this lab is having our own technology that we have developed, that is AlphaSeq technology. The strategy allows us to have targeted gene sequencing that are customized to particular diseases. This sequencing technology allows us to have extremely reduced turnaround time, that is from 24 hours to max 72 hours, and also have higher depth and coverage, having more sensitive data. While the genomic sequencing facility has been set up, equally important is the bioinformatic pipelines that needs to be set up, and the bioinformatic pipeline end-to-end has been established in our lab.
So this server basically computes all the variants that are there from the gene sequencing and gives the report back to the first entry where we have barcoded the sample, thereby merging sample entry to report generation within 24 to 72 hours. That is the currently clinical turnaround times that we are achieving. Now that you have seen the lab, I would like to apprise you of the therapies that we are kind of building here at SMSIMSR Institute of Medical Sciences and Research. There are two forms of therapies that we are currently working on. One is a combined cell and gene therapy for a rare disease called epidermolysis bullosa, and immunotherapies using repurposed commercially available biological molecules for incurable diseases. Epidermolysis bullosa, as the name suggests, is children born with blisters. Some of them die very early, less than 6 months of age, and the others that do tend to survive succumb to life-threatening cancers by the time they reach adolescence. Currently, there are no therapies available for this particular disease, and the only FDA-approved therapy costs close to around $25 billion over the lifetime of these patients. This is definitely not affordable by our patients here in India and possibly in other parts of the globe. So what do we do about that? So we are going after a therapy called revertant mosaicism. These are naturally corrected skin areas within the patient. We identify these areas using genetic testing, isolate cells, and develop bioprinted skin sheets that can be transplanted back to the patient. So this is a form of autologous, reproducible, and cost-effective cell and gene therapy. The other form of therapy that we are kind of building is immunotherapies using repurposed molecules. Basically, we identify patients with single gene defects, understand the molecular mechanism that is deranged in them, find therapeutic molecules that are already available in the market that correct these molecular mechanisms, repurpose them back, and see such wonderful results in these patients. Such forms of therapies not only improve the symptoms of the patients, but also provide better quality of life. So basically, at SMSIMSR Institute of Medical Sciences and Research, what we are trying to build is therapies for every patient across the nation and possibly across the globe, going by Sadhguru's vision that healthcare is the fundamental right of every individual.
We have a high-end genomic lab set up in this small village in the remote part of Karnataka. Our organizational ethos being one world, one family. I remember, and I want to put it on record, almost 3 to 4 years back when the medical college was conceptualized and the preparation was going on, our beloved Sadhguru mentioned that we have to do epigenetic studies in this part of the world, especially the rural population. Our foundation, One World, One Family, and SMSIMSR, on behalf of everyone, we are so grateful to Mrs. Anu and Mr. Jagadish. They in fact hail from this region. They are from Chikkaballapur, a small village called Bagalur. They have gone to US, did very well, and they are giving back to this part of the country. We are so grateful for their contribution, for their grant to take science forward and make this SMSIMSR Genomic Center a world-class center. Thank you.